Okay. So, good afternoon, everyone. It's my great pleasure to present you today, announce you the plenary of JP, who has completed, after completing a master and a PhD in the University of Southern Denmark, has completed a postdoc in Germany, Texas, California, Norway as well, and is currently a postdoc in the University of Southern Denmark, and he will now present you your Oh, sorry, University of Copenhagen now. My bad. And he will now give you an overview of his rich experience with bioacoustic during his career. JP?
Thank you so much. Yeah, I know it's a little bit confusing because it says University of Southern Denmark here, but that's just a little bit of the history about me because I grew up in a small town called Middelfart, and in Danish it's not funny at all. I know it is in English, but this is Middelfart and it looks very pretty there. But the thing is that in the waters around Middelfart, we've got the highest population density of harbor porpoises anywhere in the world. And I quickly, as a kid, found out that these harbor porpoises see their world, their murky, dark world, by using sound. And my mind was just blown. How can that happen? Of course, we are talking about echolocalization, right? But then I also realized that they receive the sound via their lower jaw, that then let the sound up to their ear, and it's just like, wow, that's so cool. And that's sort of like the beginning of my path to being a bioacoustician. Always been a geek, still, and always will. So, as Patrick said very correctly, I got my PhD at the University of Southern Denmark, where I worked with sound production in birds. We're going to get into that later. But during my studies, I developed a little bit of love for wolves. I think they're super interesting animals, so I actually found a temporary position in Minnesota where I captured wolves and put on radio collars and tracked them from a small plane. And I had so much fun over there that shortly after coming home, I found another position in Tasmania, very far away from Minnesota, where I was working with Tasmanian devils, whereas wolves are super soft and cuddly and basically teddy bears with teeth. Tasmanian devils are smelly, aggressive little bastards that you should stay away from. Yeah, I'm very objective in my description of animals. So, there's a lot of brilliant scientists that spend their entire life and maybe even get a Nobel Prize by studying something very particular, like one small protein pump in a cell membrane or some specific species or something. That's not me. And I'm never gonna get a Nobel Prize. I'm all over the place, so it's like I've been working with a lot of different species right now, and that's basically why I also had the title for this talk. Is like bioacoustics can lead you to do a lot of weird stuff with different species. That sounded wrong. So as you know, I've been working with Tasmanian devils, I've been working with goats, I've been working with cod in Norway and the harbor porpoises, also like a little bit on the sideline at SDU, the wolves, and I've done some software to analyze acoustic behavior in dolphins. And despite being not focused at all, messy, might some say, I've still been able to get into some fairly good journals. So maybe it's not that bad an idea. But I'm going to focus on a few species today because I only got 1 hour to talk about. So I'm going to tell you a little about sound production in birds because that's what I started with. I'm going to talk about blue whales and fin whales. I'm going to talk about Hawaiian monk seals. And finally, of all the weird animals, pigs in the agricultural industry. But let's start where it's all started with sound production in birds. And in order to understand, get us into the mood of sound production, I've got this video of human sound production. how we are coming down to epiglottis and down to the vocal folds. That looks like something that makes this video age restricted in Denmark. You get the idea. So is this working? Good. So what's actually happening is that what you looked down at was looking down here, right? With the epiglottis, that was the white one that you could see. That's sort of like a lid that goes in and closes off your windpipe when you're eating, which not always working. But beneath that, we've got the vocal folds, as you saw in the video. And when we are vocalizing, then they are close to each other and they're vibrating. And the vibration of these vocal folds is what creates our voices. So it's pretty much like a grand piano. So the longer the vocal folds are, the lower the frequency. So like if you played a tone here, there would be like boom. That's not how a piano sounds. And over up here it's like ding, right? That the longer, the deeper. We also know this from humans that a lot of male got an Adam's apple, and that's because the vocal folds are longer, so they basically stick out of the neck. And we've got men got about 100 hertz fundamental frequency of the vocal folds, whereas women is about 140 to 150 hertz. And there's of course some statistic uncertainty here. But also the weight of the vocal folds means something, that the heavier the vocal folds are, that also lowers the fundamental frequency. In this company, there's no reason to explain fundamental frequency right now. So that's the two. Well, there's a third and very important one, the tension of the folds. The more tension there is on the vocal folds, the higher the fundamental frequency is. And that's how we adjust it. And apparently it's something that you have to practice. I haven't sung for like 20 years, and not that long ago my niece then asked me to sing a lullaby for her. It sounded horrible. So please don't ask me to sing anything anyway. But what creates these vibrations that we talked about that we could not see in the video? We all agree that the vocal folds oscillate, right, and collide. That's how we produce sound. Is it muscles that collide them? No, it's actually not. Because if you look up here, that's the contraction time of a normal muscle. And it takes like 100 milliseconds. That's like 0.1 second, right? That means like, and this is even a fast muscle then, that we can never produce a sound that's over 600 hertz, right? Because 100 milliseconds, that's 10 oscillations per second times 60, 600. So what actually drives this vibration of the vocal folds is the passage of respiratory air. This is an illustration I made that's in a textbook that's coming out very shortly. Not doing any advertising here, but just like exploring animal behavior through sound, volume 2 from Springer. There's like this cycle of the vocal folds that if we started here where it's fully closed, and this is towards the head and this is towards the lungs, then in sound production, the first thing that happens is that they start opening. And you might see that the shape of the vocal folds is not the same shape as up here. There's much larger distance towards the lungs than there is towards the head. That creates a overpressure, symbolized by the blue color. And this overpressure inside here helps open up the vocal folds. So it's pretty much like when there's heavy traffic on the Autobahn, just been driving through Germany, that there's more air in there because it's a convergent shape. So like if there's a crash here, then it has to go around all of the cars, so it get closer together. That increases the pressure. So that pressure then helps open up the vocal folds. And then we get to a fully open situation of the vocal folds, and then we get to the closing phase, where we have what's called a divergent shape. And a divergent shape just means that the area up here is much larger than the area here. And that's the case that we see with planes when they're taking off. That on top of the wing, there's a under pressure, and that sort of pulls the plane upwards. And that's the same in our vocal folds, that this pulls in the vocal folds and help closing. And that's something called the bioaerodynamical theory. And the aerodynamical comes from what we've just shown here with the pressure, overpressure and underpressure. And the myoelastic part of this, the name, is that the reason why there is this cycle is that the muscular tissue, this vocal fold tissue, has elasticity in it. So that's a very short explanation of how the vocal folds functions. So to say it in another word, we have two vibrational components. Vibrational component 1 and vibrational component 2. So vibrational component 1 is the one that we all know, that the vocal folds vibrate and collide with each other. So they're open and they're closed. And what I've just explained now is the vibrational component 2, that they can be a divergent shape and a convergent shape. In order for any sound production to be named meet the myoelastical aerodynamical theory, these two components has to be present. We know what drives this sound production. We've long suspected that this is the case in birds, even though when you listen to a bird, it sounds like it's a whistle instead, right? So no one knew for sure that this was actually the mechanism behind it. So we wanted to do that. The case in birds is that songbirds to the left got two independent sound generators. So it's like if you had two larynxes with each their voice actually tuned to two different frequencies in birds that you could use simultaneously, that you could control individually. That's pretty cool, I think. Whereas in non-songbirds, like a pigeon that I've been working on a lot, and that's over here, that's why they call me Pigeon Boy back at SDU, there's only one sound generator. And this is sort of like the vocal folds that we see here. They are called the LVMs in birds. And here there's lateral medial labia and lateral labia. That's sort of like the vocal folds in birds. So for birds to make sound, it actually only takes two things. The song organ of birds are sitting within a pressurized compartment of the respiratory system called the interclavicular air sac, ICAS for short. And here we've got the vocal folds, as we call them, the LVMs. And there is no muscles needed for vocalizing in birds. You can cut the nerves leading to the syrinx and it can still make sound. Actually, a lot of birds will constantly make sounds if you cut the nerves, because birds are made to make sound. They're not made to be silent like we are. So all it takes is that it's the correct air pressure in the ICAS and the correct pressure coming from the lungs. Then it will vocalize. We use this for our PhD thesis to do some experiments where we created, well, mostly my supervisor to be honest, created this chamber where you could then dissect a syrinx, a song organ out of different kind of birds. This box we made in different sizes because we also put a syrinx of ostrich in there. That's a big one. And zebra fins, that's super small, as you know. And then we can just simulate that the syrinx, the song organ, is still within the bird. So we can control the pressure outside and the pressure inside of the syrinx. And then we can see what's happening. And we did that. And this is what we saw. This is a pigeon syrinx. And if we shine light for it, we can actually see the labia inside and we can then draw it. You want to see it again? So if you looked at the red and green line, then you'll see that as is actually a closing cycle where it's divergent. That there's more space up here than there is down here, right? And we see that during opening, it is actually convergent. There's less space up here than there is down here. So we've got the two vibrational components. We see that they collide, that they vibrate out and in, and we see this asymmetry of the shape of the vocal folds. So we proved that birds use the same physical mechanism as all mammals do for sound production. And that, of course, have big consequences for using birds even more as a model species for human vocalizations. Most parents tend to get slightly angry if you ask them if you can put some electrodes inside the larynx of their baby, whereas it's a bit easier to get permission from zebra finches. So we then continued this because this was pretty cool. So I went to Maine and worked with an engineering group over there because we wanted to see if we could then model the airflow and the kinetics of sound production. This is still a pigeon that we're looking at here. Because if we could that, we could maybe also do it for humans, because each year 3,000 people lose their voice when they are getting surgery in their larynx. If we could do a model that this surgeon then could see, like, okay, if we move this tissue, will it still oscillate? Will the patient still have a voice afterwards? Maybe we could do something really good for a lot of people around the globe. And they're actually starting to work with this medically. Of course, it takes some years before we are doing the first tiny little step until the doctors know how to do it correct way. I had some really nice... Something froze now. The previous one, maybe? I had some pretty cool animations here. Yeah, you've seen this animation before, right? That we can see how they move. And then if we look at the next, Can we start the next video? Then we see it looks very similar, right? And if we, can I go further? Yeah. We see that the fundamental frequency of the simulated vocal folds and the real vocal folds are also very close to each other, like 150 to 140 hertz. That's pretty close. Even the sound pressure that we get out of the simulated syrinx of a pigeon is pretty similar to what we saw in real life recordings. And doing some statistics, we can do some cool stuff about seeing what is the opening between the labia in both the simulated and in the original one. And we can see pretty similar patterns here, right? That it's sort of like, you see the blue color is when they are closing down to zero, so they are colliding. And we see, just like we expected from a meat, that they collide at the bottom first and then they collide upwards as they move. And we see that in both cases. So that's a PNAS paper there, right? So that's pretty nice. And a lot of the parameters also show no significant difference between the simulation and real life. So I think we've got a fairly good model. Okay, enough about birds so far. The next thing I want to talk about is that I got a position in the outskirts of Houston. And me being a space geek, that was super exciting. I could come and visit NASA headquarters there. And it was in a small town called Galveston on an island with a 30-kilometer long beach. Awesome Mexican food, good margaritas, nice people. Because we wanted to see if we could find an automatic way of detecting baleen whale social calls. But why? Well, let's start out with a bit of history first. Around the globe, there's these harps submerged in the oceans, and it's basically a listening station with a release mechanism so that you can go out and you can send a signal down to it, and then it releases and goes up to the surface, and you can pick it up and retrieve the recordings that you've got. It's basically just like a sound trap, giant sound trap. So it's the hydrophone and batteries and computer and storage device. And this is the ship that we used in Texas, and this is what you see to the left here is laid on the deck of the ship before we are going out and submerging it. They were actually put in the oceans by the US Navy to listen for Russian submarines. There's quite a lot of years now where we did not have to be concerned about the Russians. Have changed slightly right now. But all these years, we could just use them for listening for something useful instead. If you ask me, whales are way more interesting. So why do we want to detect these baleen whale social calls? First of all, there is no automatic detector as it was before we started this because they are more variable. So you cannot use traditional methods like spectrogram correlation, match filtering, stuff like that. So that makes it interesting, right? That we also want to detect these types of calls. Also because these variable calls are also emitted by the females. All the automatic detectors that was out there could only detect songs that's very stereotypical, and songs are only emitted by males. So we had no idea if there's a big difference in the migration routes and the population trends between males and females. So I think it was pretty essential that we tried to develop this detector. So because of the variability, I got the idea, all by myself, that maybe deep learning could be a good idea. Everyone here, of course, knows that a deep learning network is really good at classifying images. So it can quite quickly say, this is a cat. And this is back in 2019, so that's like Stone Age when we are talking about AI, right? What we thought was that spectrograms are also images. So could we train a neural network to say that this is a D-call, a social call from, and this is how it sounds. Can you hear it? deep sound. Two more coming. And now this one comes. That's pretty good sound. I also had a sped up version if the speakers could not handle those low frequencies. Just increase the sampling frequency with 5. So, what you need to do when you're using a neural network is you need to train it, right? And often it's a question about showing it a lot of examples of whatever it has to learn. So in this case, where we have to train it to recognize between a cat and a dog, we just show it. That's me teaching, by the way. You see the hairdo is perfectly fine, right? So that's how we train it, and that's also what I did with the whales. That there's a data set out there called the DCLDE 2015 data set from California. Someone's nodding their heads that know it. With annotated data set from outside California that you could use for training neural networks like this. So what I ended up with was that I actually used 2 networks. I first used a region-based CNN, and that's a network that's specialized in finding where in the image is what we want to find. As I said, this is not just a bad excuse, but this was pretty much Stone Age when we were talking about this kind of networks. So you could not get any pre-trained networks that's region-based. So I had to create my own network and train it all from the bottom. So the accuracy was not that good, but that did not matter because I just wanted to detect all regions where there could be a call. And then afterwards, I could use a CNN, a pre-trained CNN, ResNet 18. That's, I cannot see where he's sitting. Someone talked about ResNet 18 earlier today, using that. And then we could get some pretty good data out of it and can tell us there was a blue whale there was a fin whale there at that time, and there's like 78% chance that it was a whale. Because our data set for whales are huge, I did some small modifications to increase the speed of this network. So first of all, as I said, I had a custom-made region-based CNN that was fairly shallow, so it had a lot of false positives, but that did not matter because we had the CNN afterwards. But also the anchor boxes, that's the regions that it's looking for. Where is what we're looking for? If you're looking at an image of sailboats, then these sailboats can be at different distances from the camera, right? If it's close, then it's pretty big. It's further away, then it's a bit smaller. And if it's really far away, it's really small. And the neural networks that's been trained for this kind of images need to be able to find all these different sizes of anchor boxes. That's not the case with a spectrogram. The calls are always at the same distance from the camera, right? That it's pretty much same size. Of course, there's some variation, but it's not nearly as we see with the sailboats. So you can go in and then you can ask the neural network to, you don't need to look at all these different kind of sizes. You just need to look for kind of this size. And that also increases the accuracy and it increases the speed. And then finally, got this animation that they're the same size. And finally, as I said, use ResNet 18 instead of ResNet 50, also because of the speed that the ResNet 18 was much faster than ResNet 50, and we saw only slight decrease in accuracy of the two networks. So we ended up with 2 detectors. Of course, these should go all the way up here, be located up here. That's not the case, but it's still the first practical detector for detecting the social calls from blue whales and fin whales. And Scripps is, I'm working together with Scripps still on like moving forward and improving the situation all the time. I've been doing workshops with Scripps, and whoever is into marine mammals know that I was like It's basically Cambridge for marine mammals, right? So I think that was pretty cool. And third animal. Yeah, I've got an entire hour. I need to do some different species here. It's the Hawaiian monk seal. It's highly endangered. And I'm working together with University of California in Santa Cruz, where I'm also a research associate. And for these Hawaiian monk seals, we would like to create a detector, and we want to investigate the call types that they make. And we also want to look into, that was mostly me, investigate the syntax of the vocalizations. In 2021, Gillian Sells, I don't know how many of you know her, but together with a lot of brilliant people and with Lene Heismuth, like one of the giants in bioacoustics, made this paper about that Hawaiian monk seals make underwater vocalizations. And they found 6 different call types that they produce. And they recorded it from 2 individuals, Hoa and Koa. And that they also found that 90% of these calls are produced within a bout, that they make different calls close after each other. So it's like basically a sentence that they are making, right? And there are some examples of the calls here. There's a croak and a groan, growl, moans, rumble, and very short whoop. And this is an example of a sentence that they are making different kind of calls in a row. They manually took 2 days out of each week and then annotated for 1 year, the data from 1 year. But they had 2 years from both animals. So we wanted to see if we could go from those 2 days a week to everything. So basically 4 years of data that we wanted to analyze using the detector. So first of all, we had the annotations from this paper that then I had some students in Norway improve on, and then I made a graphical user interface. I'm a MATLAB geek, so everything I do is in MATLAB. I'm that old, yes. So I made this graphical user interface, and then Gillian Sells went through these 4,000 annotations that we had from the published paper, and actually found 1,000 extra calls been missed in the 2 first rounds of going through this data. That's pretty mind-boggling, right? That was it 25% of the data was missed after 3 rounds of going through. So that tells you about manual annotations. So we found these calls and could annotate the start and end of them, because then we've got a really good training set for our net. And for neural network, I chose a YOLO network. The specific about a YOLO network is that, first of all, it's a region-based CNN, so it detects and classifies. And why did I do that? Because 90% of the calls are in sentences. So they're very close to each other, so we need a network that first finds this is where it starts, this is where it ends, so that we do not mix all the different calls together in the sentences. But YOLO also got another cool thing, is that it's got 3 heads, it's called. And these heads determine what kind of anchor box, what size of region of interest that it's good at. So what we did was, because these calls are very different, a growl can last up to 20 seconds, and a whoop takes like 0.5 seconds. So it's very different sizes of anchor boxes needed for these different type of calls, right? So we could say that, okay, the croaks that are sort of like medium, a couple of seconds long, we can set that to 1 head, and then we can set the ground, the really long ones, to the 2nd head, and the super short calls to the 3rd head, and still keep really good accuracy in this way. So after some modifications and a lot of training, and I'm on a Carlsberg Foundation grant, you know, the beers from Denmark, not making Tyson here, but they gave me good money for building my own computer, so I made a really nice computer and got it up and running. And after 8 hours, I think it was, when I finally got the right hyperparameters, we actually got 95% accuracy on 10% of the runs, that we saved 10% of the data that the network had not seen. And what we get out is annotations like this. So you can see there's a growl here that the network says, okay, here the growl starts and here it ends. So we could go through all 4 years of data in 13 hours. And it had taken years for that paper from 21 for them to go through the 2 days a week for one animal. But does it match up? Do we see the same on the complete data that they did manually? Because you know you can test a network and it looks like it's working fine, but you're always a little bit hesitant. I also got imposter syndrome. That's like, did I make a mistake somewhere? So we looked at what about the distribution of the call types among these seals. And if you look, for example, at grown, 3%, this is the published data, and this is what my automatic detector found, 19, 21, 16, 18. There's a little bit difference here. But in general, you can see there's like a pretty strong image of that it looks like the same in both of them. And then we also looked at temporal trends. So this is the published data with the 2 days for the same animal, and then just 7 days a week. And you see there's still the same trend, but of course this is higher because there's more days now to count. But it all still looks like the AI matches the manual annotations. Then I look to the syntax. And this is a pretty weird plot. Sorry. But these spouts, these sentences that the seals can make, can be different length. And we wanted into like these 6 call types that we've got, where are they located within the bout, within the sentence. So I made a plot like this, because then you can go in and say, okay, we can say croak, and then we'll say for a sentence that's like 10 calls long, how is the distribution of the position of croak within that sentence? And that looks like it's pretty much even, right? The croak can be anywhere. Whereas the groan, if you go in and say like, oh, we can take 15, that looks better. Then it's mostly at the very end. If you go down, it's 15. So groan seems to be like a period. It's like when you are on the phone and with your old grandma, it's like, yeah, goodbye. That's it, right? Whereas what I think also is pretty interesting is the moan. That if you look at this structure, it looks like there's sort of like a line going down here with a bit higher color, right? So it looks like it could be a call that's sort of like, once again, when you're talking with your old grandma, it's just like, well, I'll also bet you're trying to end the conversation, but it's not the last word. But it's like a sign that the sentence is ending. And I think that's pretty interesting in a seal. You know, we've not seen this before, that there's definitely some structure to the syntax, right? We also looked into, is there some bias in the transition? So given that the seal emits one call type, is there some bias in what's the next call type gonna be? And it looks like, first of all, when we look at this matrix, it looks pretty symmetrical along this axis, right? So that we see that if the first call is a croak, quite often the next one would be a growl. And if the first one is a growl, quite often the next one would be a croak. And it's very got higher bias here. And it's also the case with moan and rumble. Rumble, moan. I think that's pretty interesting. Our hypothesis is that maybe these calls are related in somehow in some n-dimensional space, but this is not even published. This is brand new stuff that I'm just showing here. Patrick had the good advice that never talk about something that you're not 100% certain about. I'm not certain about this. Another thing that we also think is pretty interesting is the circadian rhythm, especially with the groan. As I mentioned, we had two animals, right? We have Hoa and Kekoa, and both of them apparently really like to make groan calls in the afternoon. Why? We have no idea. But there's also like, and my dog is dreaming. But also other tendencies like in general, quite often around midday that we see some slacking. Could be something with the feeding, I don't know. But let's go to the last animal because I've got 10 minutes left. Pigs. I know I was part of a documentary called If Pigs Could Talk that's been released across Europe. How many have seen it? Oh, not one hand, come on. Just give me a pity hand, someone. Well, it's been released via Arte in Germany, in Switzerland, and France. So you can probably see it online because, I'm a bioacoustician and it's not always about marine mammals that I'm working on, but just pigs in general. And we know that pigs in the agricultural industry, there is some animal welfare issues, right? So let's talk a little bit about animal welfare, that it sort of like relies on three pillars. So there's the basic physical and health function and functioning, right? That you have to, in order to be happy, you need to be fairly good condition and health, but also the emotional state if you're living in eternal fear or being stressed by someone higher up in the hierarchy, like we all know from our supervisors, but also an ability to express normal, species-specific normal behavior. You need to be able to do that to be happy. And if all those three things are present, then we can talk about good animal welfare. So how can we monitor the mental health, the emotions of the animals? So let's first start with a framework. Elodie Briefer that I'm working with, I think at least one of you, she mentioned, had been a master student with her. Was that you? I was supposed to say hi then. There's valence, there's negative valence, that's all the unpleasant things that an animal can be subjected to. That's decreasing the fitness of the animal. And that's something that the animal would like to avoid. Whereas we've got positive valence, that's all the things that increase the fitness of the animal, that the animal would really like to get more of, like treats, for example, or belly scratches, butt scratches. But we also got, on the other dimension, arousal that goes from low arousal up to high arousal. That's both for the negative valence and the positive valence. And that is defined as the bodily activation, that how strong is the feeling. So we've got negative and positive, and then how strong the feeling is, this two-dimensional parameter space. So how are they expressed, these feelings then? Well, we all know about facial expressions. You can probably see on me that I actually like talking about this, despite that it's an entire hour's worth of talking, and enjoy being here in beautiful France. And you can probably also see on my dog quite often that he's pretty relaxed, especially when he's dreaming. And facial expressions and bodily postures. But a lot of studies, as all of you know, no doubt, shows that the vocal expressions of animals can also tell us a lot about the state that the animal is in, right? And some of the most important parameters is the vocalization type, the rate, and the structure of the vocalizations. But especially the frequency means something in many cases. You all know that quite often, not always, far from always, but quite often higher frequency is something associated with negative valence, whereas lower frequency can be something that's associated with positive valence. So in 2021, that's the second time I mentioned that year, we published a paper with classification of pig calls produced from birth to slaughter based on the emotional valence and context of production, and got a lot of media attention on that one as well. You see, I'm far all the way down here. So it's Elodie that I'm still working with today. I was in charge of it. This is a director that's gotten a lot of prizes, a Danish director, Miki Mistati. And he read this paper and is like, can we use this in real life? And contacted us if we could be interested in being part of a study in real life to see if we can classify the pig calls using AI from real agricultural industry. So what he did was that he found 4 conventional farms, and in Denmark a 100 kilo pig is legally allowed, need at least to have 0.65 square meters. That's nothing for such a big animal, right? Or am I too subjective again? Organic farm, there's 2 major improvements here. First of all, they are born outside. So the mother pig and the piglets are walking around with their nose in the mud in the beginning, and then moved into ordinary stables where they then got 2.4 square meters per pig afterwards. That's a little better. And then we finally found 2 free-range farms where the pigs are living outside their entire life. And in those 3 different categories of pig farms, we then, Miki Mistrati did with advice from us. We did not know anything about which sounds came from which farms, so that it could be completely neutral, what we found out. So it's completely blind, the sounds that we got from them, and could we translate them into animal emotions? Could we find out if the pigs are actually happier in the free range in the conventional farm? You're gonna see the slide tomorrow, some of you as well, slightly modified. But whatever you want to train a neural network, you need to present it with what very close to that it needs to classify. So the neural network we had in the 2021 paper was a super clear, scientifically recorded recordings. So there was no noise on it, but these from the agricultural industry, there was a lot of noise. So we had to train a new neural network by simply adding more noise and doing some data augmentation and stuff. So it's like, what I'm going to say tomorrow is that if you want to run a marathon, you need to train running marathons, and you cannot just run a 100 meter. And if you want to become good at running a 100 meter, you cannot just train marathon running. So the result is actually here from conventional. The yellow one is the really, really bad one. That's the pig screaming and being super stressed. The orange one is unhappy, but it's not like really concerning about it. And see, there's a lot of the really bad ones, except for this one, 11% really bad sounds. That's not that bad, but it turns out we did not know that when we did this. But this is actually not a Danish farm. This is a German farm, and in the documentary that you're all gonna go home and watch, it actually turns out that the farmer had a really good relationship with his pigs. That he was out talking with them, and they had way more room than the law says that they are supposed to have. So it means a lot that you are actually got a good relationship and, you know, treating your animals decently in the agricultural industry. So a lot of variation between these farms, and stress level somewhere between 11, the German, the German, and 41% high stress. So for the organic ones, we actually very surprisingly see a lot of the really bad ones here in these two, right? Especially 56% of the bad calls. That's really bad. But it actually turned out a lot of variation again between 8 to 15. That this farm, Elodie was out doing a talk to the Association of Danish Agricultural Veterinarians, and the veterinarian responsible for this particular farm came down to Elodie and said, I know this farm and there is health issues there. And I think that's pretty interesting, right? That we could see it just from the vocalizations, that there was some kind of issue here, that they were not feeling well. And he said there was something about they got the wrong food so that they had back pains or something, because organic farms can move more than conventional pigs. So they need slightly different kind of foods to support this extra movement. So we know why this is really, really bad. But this was one of the things that they talked a lot about in the media in Denmark, was that there's no need to eat organic meat because they are not feeling any better because of it. But you know, we have to say these are 4 free and 2 farms. So statistically, we cannot really say anything about it. But it's just interesting. In the free range farms, basically none of the really bad calls, right? That's why I only eat bacon from free range pigs now. There's huge difference in when you see from these and then how happy these are. Yeah, there's 25 of these not so happy calls, but these calls are also the calls that they produce, like if another pig comes too close when it just found something nice in the mud. So it's not something that we are super concerned about. It's the yellow calls that is a stressed call that is associated with increase in stress hormone, increase in pulse, decrease of fitness of the animal in general. So it's definitely the lowest stress levels. And we also did some classical analysis, as we talked about, that call duration. Quite often, the shorter a call is, the more it's connected to positive valence, and the longer the vocalization is, the more negative it is. And we see a very nice, almost linear tendency here, right, between the conventional, very long calls, organic, and free range. But also the call frequency, as we talked about, there's not really that much nice tendency here, because the call frequency, as I mentioned earlier, that the higher, the bigger the chance is that it's a negative valence, and the lower, the more tendency to positive valence. So we prefer that it have a happy smile, and that's like the free range is the lowest again. Conventional is somewhere between the organic and the conventional. So the conclusion is that various levels of stress between the different types of farms. I'm trying to keep my time here. But free range pigs is the go home message, is that that's the pigs that's got the best life based on bioacoustics, but it's always also been seen in other non-bioacoustical studies. So what we want to do is that we want to improve the AI so that we can automatically pick out the different calls. So you can basically just have a small device on the wall with the farmer that then can tell you how is the situation in this stable. Is something going wrong? Are they starting to have pain or something, right? And we want to validate this tool by not only having the bioacoustics, but also including all the classical methods for assessing welfare, like stress hormones and now also facial recognition, stress detection. So thank you so much for your attention.
Thank you very much. Do we have any question?
Thank you. Hello. Thank you for the nice talk. I have a question regarding the sound production in the birds that you mentioned. One of the parameter that influences the sound is the air pressure. I wonder how about the stiffness of the muscles which control the frequencies that the birds sing. You said muscles don't contribute so much to the song, so I wanted to ask like the stiffness.
I'm sorry if I gave that impression, because the muscles are controlling the tension, right? That it's in humans there's a small muscle that's attached to a bone that connects to one of the ends of the vocal folds, and it's sort of like when you're tuning a guitar, that then you can change the frequencies by increasing and decreasing the tension of the vocal folds. And if you do the correct calculations for how precise this muscle is, it's mind-blowing. We are talking about nanometers that it takes, because people that are good at singing, not me, they can like control the frequency that they produce extremely exactly. And because birds are evolutionary bound to how good are they at singing. It's the good singer that gets the good female and can get his offspring, get his genes spread. So muscles have a huge impact on the quality of avian sound production. And that's also like in songbirds, there's 18 muscles that control the song organ. And most of them, we have no idea what they do, why there is 18, we don't know. In 2013, Daniel Düring and Kuhn Ellermans showed that songbirds seem to have deconnected frequency and volume control in the syrinx. So there's one muscle that controls the frequency, and then there's another muscle that controls the amplitude on top of the pressure coming from the lungs. It's very interesting.
Thank you.
Hello, and thank you for your talk. It was really cool. I have a question regarding like the emotional value, how it's correlated, the last one for the pigs, how it's correlated with the high frequency and low frequency vocalizations now. Why you choose like the pigs model, and if it's also you can apply it to other species?
The pig model is basically because we are concerned about the animal welfare issues there is in the agricultural industry. And we hope that we thereby can help improve animal welfare by validating another proxy that you can use non-invasively and automatically. As I mentioned, our hope and dream is that one day the agricultural industry can use this to improve the animal welfare, and maybe also like if there's bad cases, the government, not the government, but like the state can use this. Like I think also across all of Europe, if you're driving a truck, then you have to have like a register in your truck that shows that you are actually taking the breaks that you need to do, right? So it's like if there's issues at a farm, you could have this monitor hanging on the wall monitoring bioacoustic and saying like, there's something wrong here. And then the police, animal welfare police, if that ever happens, could go in and say like, we need to improve the situation in this particular location.
Yeah, but it's something like specific to pigs, or you can also apply it like to cows. I don't know, like this.
Oh yeah, this is also something
low
that
and
we
high pitch related to the emotion.
Yeah, that's a general trend across many species with the low frequency, high frequency.
Okay.
In dogs, it can also be antagonistic if it's a low frequency, and high frequency is a friendly voice, right?
Okay.
So of course there will always be differences between animals, but there's a lot of animals where we see this low and high frequency. The thing is that pigs are not the perfect model species for this at all, because they can also make high frequency calls when they are very happy.
Yeah.
So it's like,
Yeah, like a squeeze of
Yeah.
happiness.
Yeah.
Yeah.
but that's something that the neural network is not looking into the frequency, right? And you can just see like, oh, this is a call that's positive valence despite the high frequency.
Okay.
And that's why it's better than using the traditional method.
Thank you.
You're welcome.
Thank you very much. So we are running a bit over time, so I invite you to ask more questions during the break. I'm sure you will be happy to answer it, right? So okay, thank you very much to our speaker. So time for a short coffee break, so please try to be there at 2:45. See you soon.
So I'm Elise Foin, and I'm gonna present the results of my master's thesis on Commerson's dolphin ecology at the scale of Kerguelen Islands using passive acoustic monitoring. So the Commerson's dolphins are odontocetes. They have an odd distribution around the globe because they are found in two distinct populations. You have one in Argentina and one in Kerguelen Island. And most of the knowledge on the species ecology that we have for now is based on studies conducted on the Argentinian population. And what we know is that it's a highly coastal species, it has an opportunistic diet, and it shows some kind of seasonal distribution. And so now we want to investigate the behavior of Kerguelen's population. And for that, there is 3 chapter of research that was conducted on this population. You have one on visual observation, one with genetic analysis, and then the one that we are gonna be interested in is the passive acoustic monitoring. So the first results showed the same thing that we had on the Argentinian population. So the catalog and the visual observation showed that the species is highly coastal. It doesn't go further away than 100 meter depth, and it seems to have a resident lifestyle on the scale of the archipelago. And then the biopsies showed that there is a complex structure. Now we are gonna talk about what we could find with the passive acoustic monitoring. So the question we had was, does the species show any pattern in its distribution around the archipelago? And I'm gonna say you why this species is suitable for passive acoustic monitoring. So, as you may know, cetaceans emit sounds in their environment, both to communicate and to feed. And so the Commerson's dolphins are no exception. They emit narrowband high-frequency clicks that are at really high frequency and that we can isolate. And we can have 2 informations with these clicks. It's that we can have information on their presence and also information on their behavior, because we discriminate clicks between their intervals, and if the interval interclicks are lower than 5 microseconds, we can say that it's foraging behavior. The program was conducted using this kind of instrument, which is a specific instrument that can record clicks occurrences on a long time scale, because it has a high autonomy and it can record every timestamp for every click emitted in the environment. And it has a low detection range, around 300 meters. So it allowed us to isolate the 4 sites of the study because they don't cross. And here you have the 4 sites around the archipelago, in one specific site of the archipelago, which is the Gulf of Morbihan in Kerguelen. So for the data collected, we have a calendar on the 4 sites. As you can see, there is some discontinuity in the data because of mostly logistical issues. But overall, we have approximately 60% of recorded hours. And what we can use that information, we can use it by isolating every minute's positive to detection in order to analyze their presence around the archipelago. So we have two information, as I told you. There is the percentage of minutes that are positive to detection, and then there is the percentage of minutes that are positive to detection that are positive to feeding buzz. And then we compared these results with 3 time scales. We have the interannual variability, the monthly variability to show any seasonal activity, and then the dial pattern. So first we saw that there is some kind of interannual variability. The acoustic activity is not constant around the archipelago, and it seems to decrease over the years. And also you can see that the site D is really low in detections. That's for the proportion of feeding buzzes. It seems more consistent than the DPM detections. So it would seem that the foraging behavior is a proportion that's consistent. For the months variability, we have a high seasonal pattern around the sites A and B. The site C doesn't show as strong pattern, and the site D is really few frequented. And for the proportion, you have the same pattern, like it's still consistent around the month. Then we highlighted a dial pattern, a strong dial pattern. We saw that the dolphin seems to emit mostly at night and really few clicks during the day. And for that, the proportion of feeding buzzes are following the same pattern. And to compare that with what we knew about this population, we know that it's consistent with what was observed around the Gulf of Morbihan in the archipelago, and that's it's consistent with historical observation too, because that's from a study published in 1997, and we already saw that the cormorants seemed not present in the archipelago, I mean in the Gulf, around September, so it seems that there is a high seasonality. And it seems also that they are visiting other areas of the archipelago, like the Baie du Repos, which is northern on the archipelago, and it's consistent with what the biopsy showed. And for the dial pattern, that's to be compared with another species that emits NBHF, which is the harbor porpoise. And we don't have the fishes population data, but for the harbor porpoise, the prey availability seems to drive the presence of the harbor porpoise activity around their area. So we could hypothesize that it's the same pattern here with the cormorant dolphin. And so the limits are the discontinuity of the data, as you saw, and the small detection range of the instruments, because if the animal is not directly in the area of the instrument, it won't be detected, but it doesn't mean it's not in the area. And it shows only acoustic presence, and we don't know if the animal is there and silent or just not there. And for the perspectives, it would be interesting to investigate the other area where we sighted dolphins to see if they show the same acoustic activity. Thank you for your attention.
Yes. Thank you, Elise. Are there any questions on this presentation? Yes.
Thank you for your presentation. I may have missed it, if so, I'm sorry to ask the question. How did you find the clicks manually, or?
No, so the CPODs are instruments that are built by someone that also built the software to extract every clicks, and there is a classifier that is the kernel classifier, which is able to isolate NBHF from odontocytes click from both click. But here we have only an ABHF species, so we only kept the NBHF clicks.
All right, thank you very much.
Other questions? All right, I have one question. Are the visual and genetic studies still happening? Right
The
now?
visual one, yes, and the genetic, no.
Did they also find a decrease in presence, the same as you presented?
They didn't communicate about this. I couldn't find any information about the year interannual variability. We don't know.
Okay.
Yeah.
Okay, thank you. Do you have any idea or hypothesis as of why it could be decreasing?
Maybe they are changing their distribution around the archipelago, but I think we are missing environmental data to conclude about it.
Okay. Any other questions? All right then. Thank you, Elise.
Hi, I'm gonna make a short announcement. We are aware that the room is really hot and we're sorry about it. We are having an AC that's right here. We have fans for people that urgently need to be a bit colder, but also like be careful. If you feel bad, just tell us and take care of yourselves. And if you want a fan, we have fans, okay?
So now we will be listening to Ellie. Ellie, the floor is yours.
Hello. Is that working? Yeah. Oh, cool. Hi, everyone. So I'm Elie, or Elisiane, and today I'm going to talk about this project that I have just started. So this is called, I think you have some other title on the program, but this is called Ecoacoustics Balears. It's a new approach to spatial temporal biodiversity monitoring in the beautiful Balearic Islands. Oh, where is the chick chick? Oh, thank you. I'm not gonna take longer in this one. Like, why do we use ecoacoustics for scalable biodiversity monitoring? I think everyone has been talking about like why it's so great about the use of sounds for environmental monitoring, and how cool is that we have some really great recent advances in technology, and how we are processing our data much differently than we were 5-10 years ago. This is just an example of, yes. This is a study, a review that was done in 2019. I'm sure there's way more studies now if you have like a more recent review, but it's quite interesting to see that that we are seeing a lot more people using those automated recorder. This has really increased in the past 15 years or so. But when we look at which groups are we focusing on, we still have, well, there's bats here. Like I haven't seen any presentation on bats yet. Wow. But birds, of course, are the second most well-studied groups. And the invertebrates, poor guys, they are really like here on the really on the bottom of the preferences. So this is like the first thing that I wanted to highlight, that in terrestrial environments there are much more than just birds. And this is like a graph that I quickly made this morning from some data that I'm collecting for this paper that I'm preparing now. That's exactly talking about how insects are being more used in ecoacoustic studies, and how is the state of art of it so far. And it's interesting to see that while we have much more presence of or people just mentioning insects on their ecoacoustic studies, Here in blue, you have like the studies that are identifying to the species level, and it is still quite low comparing to just saying, yeah, those are insects and so what. So that's a big challenge, I think, yet, like going from bioacoustics to ecoacoustics when you talk about insects, and there's of course a lot of limitations on this, but I think we are in a very sweet spot now, like to start to talk more about insects, bioacoustics, and how to actually start to use this. And well, if you're interested, like maybe later this year, I'll be releasing this paper, hopefully. Oops. So yeah, a few months ago, when I moved to Mallorca on the Balearic Islands, the first thing that I tried to do was to find someone who was working with ecoacoustics, with bioacoustics, whatever, and there was no one. So we developed this project, me with some colleagues from there. We are developing the idea for this project that is really like to develop and implement this bioacoustic monitoring framework on the islands. With this very broad goals of identifying the soundscapes, spatial and temporal dynamics, but also focusing specifically identifying birds and orthopterans on the islands. And if you think about Mallorca, that's probably the idea that you have, and it's true. That's what we have there. I'm already homesick. But Mallorca is much more than that, and it's quite an interesting environment for this kind of studies because first it's an island, so you have like very well-defined borders there. And second, you have it's not that big, it's not a huge island, but it's not like super small, so you have quite a variability of different environments and also different degrees of anthropic impact on the island. So you have like a good system to develop like a nice soundscape map of the island. This is very illustrative. Those are not my points yet, but we are working now on how to stratify our sampling for the whole island. So as I say, like I just started a couple months ago, and we are still working our way on how many sites we're going to use. We're thinking about using around 100 sites that are going to encompass like different levels of protectedness, land use, and vegetation class. We're going to collect data every season for around 2 weeks per season, and quite interesting. I'm very interested in the 24 and continuous data, in 24-hour data, as opposed to just having the morning or night or just like short clips. Because I'm also interested in acoustic niches. I think that's what everyone thought that my presentation was going to be about, but this is going to be a bit further down the road. Just quickly going through our methods. So the first thing we are doing, so we just started collecting a little bit of data, and the first thing we're doing, it's really like pre-processing. And yes, we're doing acoustic indices for that, like not the classic ACI or whatever, but we are doing some acoustic metrics to find things, for example, like clipping in the data, or even we're even thinking about using some clustering to separate sound classes and then working more detailed into them. And then we're going to move to species identification, which of course is BirdNet with some manual verification. I'm having some great ideas from the presentation that I'm seeing here. Thank you so much for everyone that's working with BirdNet. But with the orthopterans, it's a bit different. We are working with this package that's called Orthoptera, really to help us to more quickly having a nice database of the species on the island. And we're working with local experts as well. Our idea is really to have a good data set of the taxonomic, phylogenetic, and functional diversity of birds in the island, and how does that relate with different land use and also with different seasons. Some preliminary results. So as I say, we have some nice degree of some good variation of habitats in the island. And we are trying to, for this first preliminary data that we just collected, I've just retrieved some recorders last week. So we, come on, oh yeah. So I'm gonna just present a little bit about like data that we collect on this or algahoba and almond plantations. We also have we had to also record on farms, pine wood forests, and also on coastal areas that we call the garrigas. And the first thing is that's very old school. That's a false color spectrogram. And this is the colors here, they're not reflecting amplitudes. They're actually reflecting a combination of 3 different acoustic indices. And this is quite nice. This is like the 2 visualizations. It's a 24-hour, and you have like 2 different visualizations that are built with different combination of indices. Oh, wow, we're already 2 minutes now. And from here, just by looking at that, like we can identify a few different animal groups, or sometimes just geophony and stuff like that. With BirdNet, as I say, we just started to look into that. But the first thing we notice is that there's a lot of variation in the number of detections, not just in the species that is detecting, but also like in the number of the detections for each one of our areas. So our idea is really to deal with this variance is really just to start to manually verifying and manually review and start to work with the confidence levels as everyone was talking before with BirdNet. But really to separate this also into our different habitats that we have. And for the orthopterans, now we are at the stage where we are working with focal recordings and previously collected data by some local experts, and we're starting to build this nice database for the orthopterans of the island. And then we can start to move on to try to identify this from PAM recordings. Just as a curiosity, like it doesn't sound like you're gonna discover something new in there, but actually, like just working with the recordings a couple years ago, one of our collaborators found like for the first time a species in the island. So this was a species that really looked like another one, but just by acoustically he could differentiate that from the other species. So this was just 2 years ago, and we're very hopeful that we can find something interesting there. And I want to thank all my collaborators, the guys that are working with birds, diversity, and Jaume that is working with the orthopterans, my co-team there in the island, and my little fieldwork assistant as well. That's it. Thank you.
Thank you. Any questions?
Hello, thank you for your presentation. You mentioned in one of your slides that you were using or planning to use classifying techniques.
Sorry, the classifying techniques for?
Yes, for sounds. I don't, maybe if you can...
With the birds, you're saying?
No, before.
Before. Okay.
Before.
Okay.
Before.
Is there anything before that?
Before. Yeah, maybe it's in before. Stop.
Okay.
Clustering. Yeah, sorry, it's I didn't use. What are your aims with clustering? Because you know that biological some production by biology can be very confusing. Some species can sound absolutely different to what they are. So what is your plan here?
Well, that's a good question. I have some experience with clustering in a very different environment, but working in tropical environments, and we actually have some nice results with it. But really, like classifying different sound sources very broadly. But yes, we did have some nice results with that. I think that's really a first characterization of the soundscape, especially if you're gonna actually work with 100 different sites, and there's a lot of variability, but really trying to understand like where do we have more I don't know, more activity by birds, or even just like biophony, or have my biophony or geophony or stuff like that. It's really more like a first characterization, but it works quite well with the insects again. So if you want to just for birds, yes, birds are tricky. But for the orthopterans, for me, like I remember working quite well with the clustering. Like you could see like very different clusters, like with just the orthopterans. So if it's not, we don't still have a bird net for orthopterans, so maybe that's a good way to move forward, like having at least like this first selection of the recordings that we're gonna work with.
Okay. Oh, many questions.
Thank you for your talk. It was nice. You mentioned acoustic indices. Which indices are you using?
Oh, for the visualizations.
Yeah.
Oh wow! I forgot. I can have a look for you. I know in the first I forgot because those are 6 indices. I know this red one here is like a background noise index. It's called, and there's a pardon me. This is an ACI. There's also a background noise index that it's not being showed here. Yeah, this red one I'm sure is ACI, but the other ones I'll have to check to say. But who developed this? This was a group in Australia that developed these visualization techniques, and those are the standard indices they are using for it. But it's quite flexible, so this can work for marine environments as well. It's yeah.
Okay, thank you.
Just a quick question. I don't know the endemism rates at Mallorca because it's a specific situation. It's an island, etc. Do you plan to deal with this specificity of location and size of island? It can be change the sounds that are recorded, that are used in BirdNet or etc.
Sorry, you mean like the variation in the calls used by BirdNet in the island?
Yes, BirdNet is built around databases that I think maybe there is not so many songs from Mallorca. Maybe Mallorca it's something special, and maybe
that was the first when I presented the project. That was the first question of one of my collaborators, because he said that because the BirdNet was trained with data from other parts of Europe mostly, we could see some variation with the data from Mallorca. From our first rounds of reviews, like it's still looking very promising, but I think that's why we are working. We're still gonna go through a long process of manual verification, and we have a few experts that are gonna work with us on that. But that's a good question, because we might end up not having such a good results as in other areas, of course.
Thank you. And next, and for the final presentation.